Hello, everyone. Nice to meet you today. I'm delighted to present you this, to introduce you this webinar regarding the latest trends, testing trends we have seen at the IFSCC Congress last week in Cannes. First of all, we will have the pleasure to get the presentation of several actors of the testing field. First, we will start with Loredana BIRCA from CIDP for 7-8 minutes each. Then Julien PRZBYLSKI from Pixience. Then Pauline COTINAT from Innov&Sea. Nima ASKARY from Scibase, and Julien CHLASTA from Biomeca. After this, I will present you an overview of what I found very interesting about testing at this Congress last week. So let's start directly with Loredana. I will stop sharing my slide, and please I let you the floor.
Can you see my screen?
Yes.
Thank you for the introduction.
And I just forget one thing. Everyone who wants to ask a question, there is a chat, and we have always an eye on the chat. And so after each session, you can ask your question, or we can wait also the end of all the presentation, and we can have a round table about the question you have. Thank you.
Thank you. Thank you for the introduction. Today's topic will be SPF testing, the new in vivo and in vitro methodologies. I will start with a small presentation of CIDP. We are a full-service CRO. We offer our clients preclinical research, clinical research, biostatistical and data management, scientific and medical writing. CIDP is a global CRO with centers in Mauritius, India, Brazil, and Romania. Since this session is about photoprotection of cosmetic products, an important point is to mention that this type of studies are exclusively done in CIDP Romania, in our affiliate from Romania. Our center here, it's located in Bucharest, the biggest city from Romania. CIDP Romania is registered with FDA. It is an important point for our clients that want to put their products on the US market. Also, we have CRR accreditation is important for our French clients. CIDP Romania is an active member of AFNOR, and also is participating in the BIPEA with very good results. Beside the photobiology studies, also we perform safety studies, efficacy studies, medical device studies with across the group with over 20 years of experience in clinical trials. We perform around 80,000 studies until now. As I mentioned before, our main field here in Romania is on photobiology. Here on this slide, you will see a list of type of studies that we perform, and also a type of stress claim that we new developed. And I will start with the first one, the daily routine resistance. That is a claim that we the objective is to ensure continuous sun protection without compromising skincare or makeup routines. As you already know, all skincare or makeup routines are consisting in more than one product. So it's important for the clients that provide the sunscreen SPF to be able to demonstrate that the level of protection of the product is the same, even if you more products. The next claim is the hairy areas protocol that ensures hairy areas receive the same level of sun protection of the rest of your body. As you all know, the body has different parts of the body that are covered with hair or not, and we are very happy to be able to demonstrate that the product can protect the same even if it is applied on hairy areas. The resistance under the extreme conditions, this protocol is developed, as you know, the climate is changing, is diverse, so we want to make sure we can demonstrate that the product has the same level of protection, even if you apply it on humidity, on intense sun, on different regions. The sweat resistance of a cosmetic product. Other stressor that can impact the efficacy of a cosmetic product is sweat. So we can demonstrate by applying this process that you have described here in this slide, and demonstrate that the product can protect even if it's applying an sweating process. We use a treadmill to perform this activity. Salt water resistance, this protocol was performed to better stimulate real life conditions like salted water from the seaside. Also, it's applicable for the chloride water resistance protocol, can stimulate the real chloride water from the pools. Both salt from the water or chloride from the water from the pool can be stressor that can impact the efficacy of the product, the level of the protection. Using these protocols, we can demonstrate that the product can or not protect even when they are applied in these conditions. As you already know, there are two new methods for SPF in vitro testing, the double plate method and the HDRS method. The double plate method is ready to be performed in our center here in Romania. This method is not only enhancing reproducibility and ethical compliance, but also streamlines product testing for emulsions and similar formulations. Also, offers a faster alternative to human testing. The HDRS method will be implemented in Romania on upcoming year. It offers a modern and non-invasive method for determination of the level of the product, of the protection of the product without inducing erythema. We are very excited to be able to provide to our clients these new methodologies. As CIDP is committed and follows all the legislation and guidelines for all our clinical studies. We use the FDA regulations, the good clinical practice, the ISO methods. Also, you have on this slide the key equipment that we use for testing our products. So thank you very much. For any clarification or more talking about stress claim, you also can find us on Cosmetic 360, and also you can look for us on LinkedIn. Thank you very much.
Thank you very much for your presentation.
Thank you, Loredana. We have a question from Patricia Garcia.
Yes.
In the
Yes,
chat.
please.
She will speak.
I'm sorry, Gabriel. Can you ask the question?
Yeah, normally, Patricia Garcia, you can speak and ask your question if you want. Okay, I'm sorry, it may be a mistake.
Okay. Great. Thank you very much, Loredana.
Thank you.
I will introduce you, Julie, from Pixience. Julie, thank you very much for your presence today, and I let you the floor for your presentation.
Thank you, Anne. Okay?
Yes, it's okay.
Perfect. Thank you. Good afternoon, everyone. I'm Julie from Pixience company. Thank you all to be here. I hope you enjoyed the show of IFSCC last week. Today, the goal of my presentation is to explore with you the C-Cube imager versatility to quantify and visualize the effects of different products. So if you don't know our company yet, Pixience is a pioneer of microdermoscopy. We develop C-Cube imager in France for laboratories and CROs around the world. The C-Cube imager is the key system that can combine the instrumental rigor of probe system and the visual power of an imager. This tool allows you to have a complete chain of skin analysis system. The specificity of this device lies in its ability to target a specific area and perform a precise measurement of effectiveness on it. And this with a very little error margin of only 0.01%. So C-Cube is also variable in any room light, maximizing user comfort. No need to be in a dark area. In addition, it is calibrated colorimetrically and metrically, which make it possible to obtain consistent results over time for different analyses and different sites for multi-center studies. In terms of claims, C-Cube can cover nearly 90% of needs for studies from in vivo to ex vivo and also hair analysis. Its ergonomics, only 400 grams, allows it to be flexible in the areas of use, and it also works on all phototypes. So it is a very versatile tool. For the technological part, C-Cube is based on 3 fundamental points. First, a reproducible color calibration correlated with chromometer. Secondly, perfect optic and indirect lighting ensure a consistent image without distortion and hotspot on the center. And the third point, it's the true 3D reconstruction by the technique of stereophotometry in accordance with ISO 25178, allowing calculate 3D parameter like SA, SQ, roughness parameter. Let's take a look to show the importance to have standardized and homogeneous images for both color and surface measurement, particularly for pigmentation spot measurement. With the C-Cube, the delta E color difference across the image is very low, and you can see on the right that it is the same for surface variation. It means concretely that data is reliable on the entire image, edge to edge, and it is essential to truly assess the effect of the product or the active ingredient and without having a bias from the device you are using. For pigmentation measurement, C-Cube calculates pixel by pixel pigmentation index based on the concentration of melanin in skin. The results can be displayed in color maps with export of minimum, maximum, and average values. So this able to evaluate the pigmentin claim, whitening claim, and so on. And you can see on slide a recent example of the use of C-Cube for pigmentation, a study of the beginning of the year. It aims to evaluate the therapeutic and preventive effects of lead treatment on the post-inflammatory hyperpigmentation and also erythema. The both calculate with C-Cube. For the software ecosystem, C-Cube can accompany you all along a project, starting from the first idea in research. This allows you to have the same base of measurement on the entire project. So first you have the quick scale software for upstream phases, the panel inclusion, the research and development and pre-testing phases, and the clinical research of software for testing phase, analysis, and marketing exploitation. Specific and important point we have recently developed at Pixience, and we continue to work on it, hair analysis. Historically, we've been working for many years on skin image processing. That's our heart of job. But today, thanks to artificial intelligence, we can go further and detect some elements in image, and we know that this is particularly interesting for the hair application. So C-Cube was already able to detect automatically hair density with follicle, but now it can also detect hair diameter and length in an entirely automatic way. So this new automatization make it a complete and user-friendly tool for an entire hair study, for example, for a regrow product or a thickener product. So to sum up, I would like to quickly remind the scope of C-Cube abilities. So it is an imager for 2D and 3D, also a hair analyzer with more and more automatic analysis, and it can also be used to perform ex vivo and in vitro analysis with patch, skin explant, etc. So many publications has validated C-Cube performance. You have some example, recent example here. And performance has also been validated by our customers, laboratories, and CROs. And to end the presentation, we can say concretely that C-Cube fits easily into many types of clinical studies. It allows to validate wide range of claims through 2D, 3D, hair, or even ex vivo and in vitro analysis. So it is a tool that stands out for accuracy, reliability, versatility, and ease of use. Thank everyone for their participation. Thanks Skinobs also for this opportunity, and don't hesitate to ask your question.
Thank you very much, Julie. I will just check the chat, and it's okay. We don't have any specific questions. So now let's go after Julie's presentation. Let's dive into the coral reef with Pauline. Pauline, I let you the floor.
Okay, thank you, Anne. Thank you, Anne, and thank you all the Skinobs team for organizing this webinar. So let me share my screen. Okay, should be okay now. Perfect. If nobody tells me, that's everything okay. So hello everyone, my name is Pauline.
Okay.
Okay, perfect. Thank you, Anne. So my name is Pauline. I'm glad to be here today to talk to you about preserved marine ecosystems. So what I will start with is just like quick presentation of Innov&Sea. We are the first laboratory to offer in vitro ecotoxicity tests based on cnidarians. So we are based here in Nice in the south of France, and we are specialized in the study of marine invertebrates and their response to stress and their cell cultures. So we launched our technology on the market in early 2023 with the different supporters written here. But everything, all the story started with an observation, a dramatic observation. More than 20% of coral reefs have been irreparably destroyed in the recent decades. So it showed that the need was for tests to protect these marine ecosystems in danger and to help the cosmetic companies to seize this topic and to make their business more sustainable. So what we saw is that we were not the only one to have this observation. All the consumers started to be really conscious about their environmental impact. They started to think about the ocean-friendly products when they have to choose a sunscreen each year. But also, more importantly, the authorities started also to increase pressure on the cosmetic industry. For example, some UV filters were banned, starting in Hawaii and then in many different countries. And now in Europe, new regulations appeared on environmental claims, especially governing scores and labels on the packaging. So basically, when you are a brand now, you are stuck between these two problematics, the consumer needs and the authorities' pressure. But moreover, you have to stay competitive to address all this demand, to support your eco-responsible approach, but to continue to have this challenge to develop efficient sunscreen products with effectiveness, but more respectful for the marine environment. So what you can do when you are a brand is to do ecotoxicological tests. It's not easy to say. The classical method to do this kind of test on marine environments is to use living marine organisms, such as coral nubbins, crustaceans, or even larvae of sea hurricanes or fishes. And to test, basically to put in contact with the substance you want to test and to see if the model will die or have problems with the reproduction, for example, different kind of things you can see. But what is really what was really important for us is that all this approach have some limitation, technical limitation with the reproducibility of this approach, the cost also. It can be really heavy to implement to have these huge aquariums, etc. But more importantly for us, it was a consumer perception, because to verify your product is not toxic, you have to basically destruct living marine organisms. So at Innov&Sea, we asked, what if you can have an effective test, but without any animal sacrifice to meet the consumer expectation? So we went back to what we wanted to protect at the beginning, the coral reefs. So we went to this family of animals called cnidarians. Cnidarians are the corals, the sea anemone, the gorgonians you see when you dive close to the coast. They are marine organisms, but they are really threatened, endangered, and we want to protect them. So in the lab, we started to study one of these organisms, a sea anemone, Anemonia viridis, that is a recognized indicator for these coastal marine ecosystems that is in the same class, in the same family of reef-building corals, and also, just as corals, they live the bleaching phenomenon, and they live in symbiosis with a microalgae. So it was the perfect model to illustrate these coastal ecosystems, but also because on this animal, we developed an in vitro approach. So in fact, we took some tentacles from the animal. They regrow. They have a huge regenerating capacity. And with these tentacles, we extracted the cells, the animal cells, but also the microalgae that are inside the cells to do some cell cultures and totally in vitro technology that we could then preserve, stock, have like a biobank, and also used for biological analysis. And that's what we do now with this in vitro technology. We basically test different kind of products. It can be raw materials, ingredients, UV filters, preservatives, you tell, but also formulation blocks, more complex products, and in the end, the final products. So what we do is to test the toxicity of the product by putting in contact with the cells to see if the viability and if the cell growth are maintained or not, depending on the product. And then we also see if the impact we saw is reversible or not by adding a resilience phase. And with all this data, we can deliver a grade, a score to each product, so that can help you to compare easily the different products you want to use, the different raw materials, or improve the formulation of your sunscreen product. So this approach is made to reduce the use of animal testing, but also this approach gives many more advantages, such as a high reproducibility and a precision with the attribution of a score, a very precise score. It's available all year round, and it's really fast. In 14 days, you have the result. Is it suitable, as I'd say, for every step of the formulation, from the raw material to the finished product? But also what is highlighted here is the eco-responsible approach of this test, totally in accordance with the REACH regulation and the United Nations Sustainable Development Goal, to avoid, in the end, the use of living animals for your test. So we just to come back to what I said at the very beginning, you saw we all know now that this kind of allegation, coral safe, ocean respect, they are too globalized. I don't know how to say in English, but they are now bound. You cannot say this kind of things. So our goal in Innov&Sea is to help you to optimize your claims, to find the right terms, the right phrases with scientific arguments to communicate better your eco-responsible approach to the final consumer. So I want to thank you all for your attention, and I want to thank again the Skinobs team for this great webinar. Thank you.
Thank you very much, Pauline. We had a question. We have a question now from Nikita Radionov, who asked if it's if your technology is compatible with testing oily product or creams.
Yes. Yes, sure. Our technology is compatible with any kind of formulation of Galenix. We tested, in fact, creams, oils, stick, and even different kind of raw materials. So it's really adaptable to every kind of product.
And we have another question from Simona Steiger in the link with this formulation, oily formulation. And how do you apply final products, oil water emulsions on the marine cells? And will it be diluted with a solvent or solubilizer?
Yeah, exactly. Yeah, the product can be solubilized. It's hard to say. They can be solubilized, in fact, to be compatible with the water-based medium for the cells. So if needed, we can use some solubilizer that are already tested for the impact, of course, but to improve the test, of course, yeah.
Thank you very much. Thank you very much for your question. And now, thank you, Pauline. And we stay online until the end. And now I will introduce Mr. Nima ASKARY from Scibase, clinical director and project management for Scibase. Nima, the floor is yours.
Thank you. Thank you for having me, and thank you for Skinobs for this opportunity to present our technology. Today, I will talk about a technology that we believe will greatly improve the ability to assess and quantitate skin barrier function moving forward. It's a technology that can be used non-invasively at point of care to provide information about the subclinical status of the skin. The journey of Nevisense began more than 35 years ago at the Karolinska Institute here in Sweden, with Professor Stig Olmar setting up the very first experiments. From those beginnings, the research has grown into today's clinically validated Nevisense platform that is shown in the lower picture. Since then, EIS has been featured in more than 85 peer-reviewed publications and over 5,000 cases documented. Nevisense has a clinical indication for skin cancer. It is an FDA-approved device for melanoma detection and a CE-marked product for both melanoma and non-melanoma skin cancer. Nevisense is now being increasingly used in research to assess skin barrier function. Currently, we have more than 35 ongoing studies worldwide. These studies cover areas such as dry skin characterization, evaluation of pharmaceuticals, cosmetic testing, with endpoints and identification of skin type and treatment responses. Here's a list of the different fields where EIS is applied in different areas of skin research. You can see skin irritation, hydration, lipid content, and UV stress. All of these directly linked to cosmetic product testing. There's also strong data in atopic dermatitis and pediatric skin-related disease, which we'll go through. So I just wanted to briefly introduce the core of our technology, which is electrical impedance spectroscopy. Nevisense and EIS works by sending small electrical signals into the skin at different frequencies and measuring the tissue's response. The signals that this electrode sends through the tissue generate about 700 data points per test. These signals reach different depths in the skin, up to 2.5 millimeters, meaning that we capture not just surface level information, but also deeper structural and compositional information. The signals are influenced by the tissue structure, its hydration, and moisture dynamics. Larger data sets can also be processed using AI tools, which interpret the complex signals that we just mentioned and generate scores that are easier to understand. In simple terms, EIS provides a non-invasive way to look at deeper into the structure and function of the skin. This example shows how EIS can detect differences between atopic and normal skin, even in areas that appear healthy visually. The signal reflects many factors such as tissue structure, organization, cell membrane integrity, cellular orientation, tight junction integrity, and moisture compartmentalization. If you look at the graph in the example, there's a clear difference in the profile of the impedance curve between the normal skin and the visually unaffected skin of a patient with atopic dermatitis. Now that we've explained the technology, let's go through some of the studies where this has been applied. One of the first questions asked when exploring atopic dermatitis was, can EIS detect barrier change even if the skin appears unaffected? This slide shows a Swiss study on patients with atopic dermatitis. It describes the ability of Nevisense AI algorithms to differentiate between skin of healthy controls and normal-looking skin of patients with AD. On the left, you can see the EIS score. There's a clear and significant difference between the healthy control skin and non-lesional AD skin, which shows that EIS is sensitive enough to detect the subclinical changes that are not visible. which is highly relevant, not only for dermatology, but also for cosmetic and therapeutic product testing. This slide is from the same study where we can see that how EIS and transepidermal water loss performs when comparing healthy skin, adjacent skin, and normal skin in patients with atopic dermatitis. On the left, again, you can see the EIS results, which clearly separate all three groups. So healthy skin, adjacent skin to lesions, and lesional skin, which demonstrates that EIS is sensitive enough to detect subclinical barrier damage, even if the skin looks normal. The clinical implications is important also for cosmetic testing. It shows that you can detect early barrier changes and monitor the spread of barrier disruption before it even becomes visible. Here you can see another slide from the same study. EIS can also be used to monitor the skin response to treatment and healing over time. Looking at the graph to the left, we can see that within a 3-week period, the patients showed a clear improvement in their EIS scores, quantifying barrier recovery in a way that is objective and measurable. What's also important is that these improvements correlated to the clinical severity measures, such as PRZBYLSKI and SCORAD. As you can see, interestingly, one of these patients did not respond to the treatment, which was also observable by the EIS score. On the right, you can see some curves where EIS shows a strong inverse correlation to inflammatory biomarkers. This strengthens the case that what we are observing is truly reflective of the underlying disease activity in the skin. So in other words, EIS doesn't just detect barrier damage, it provides a way to monitor treatment and response and recovery to a drug or cosmetic product. Here is a case study from Turkey where a young child with very severe atopic dermatitis was started on a biological drug and followed for 5.5 months with repeated EIS and SCORAD measurements connected. On the graph, the red lines show 2 different EIS results. These are not AI algorithms, but specific data points out of those 700 data points that I mentioned previously. And here you can see a strong parallel. As SCORAD decreases, indicating clinical improvement, the EIS score also changed, showing barrier recovery over time. Yet another publication that shows the EIS can objectively and reliably monitor treatment response. And the last publication I wanted to mention has EIS as one of its main endpoints. Here, the study examined how various everyday factors can interfere with skin barrier measurements. Transepidermal water loss, which measures water loss of the skin, is known to be sensitive to a lot of environmental and behavioral influences. So on the left, you can see that transepidermal water loss was significantly affected by the study participants climbing a set of stairs before the measurements were performed. On the right, you see a similar setup, but with EIS, where the EIS measurement remained stable across all time points. So neither physical activity, caffeine intake, or skin washing impacted the EIS results, which shows that EIS provides a very robust and reliable way to assess skin barrier function, which doesn't have those confounding factors that we're used to when assessing skin barrier function. So in summary, Nevisense or EIS is an established technology based on studies conducted at the Karolinska Institute. It is clinically validated and being used as a clinical tool in multiple countries around the world. Studies have shown that it has a robustness and reliability that can make it a useful tool when assessing small changes within the skin barrier and very sensitive to minute differences within the skin barrier. So Nevisense is an advanced and innovative method a strong complement to transepidermal water loss for assessing skin barrier function. Thank you for your time.
Thank you very much for your presentation. We have some questions for you. One from Robin Dumas, who asked, is it possible to work with your product on the diversity of presence of microbiota of skin areas, basically looking at the microbiota?
There are a number of studies where comparing the EIS score to the occurrence of different microbiota in the skin are being assessed at the moment. So we do not have the results at the moment, but there are theories which have indicated that this is an avenue to go down. Absolutely.
Okay, thank you very much for your clear answer. There is another question from Małgorzata Tarnowska, who asked, which classes of inflammatory biomarkers can be tracked with EIS, and are they limited to the ones you showed in the study?
There are a number of biomarkers. I do not have off the top of my head the exact ones, but the ones that I showed on the screen are not the only ones. They are the ones that were presented in the poster, which was the most significant ones. But there are a number of biomarkers that are.
Thank you very much. And a last one from Annick Foligné, who asked, thank you very much. Can EIS be used on skin explant to measure effect of products for cosmetics on explant?
We do have ex vivo measurements. For the sake of time, I actually removed the studies that we have on explanted skin, but we do have publications that show that when Nevisense is used on ex vivo skin to determine detergent effects on the skin barrier, there is a very clear dose response effect being identified when looking at these ex vivo models.
Okay. Thank you very much. We will close the question now, and to keep the time okay for everyone. Thank you very much, Nima, for your presentation, and please stay until the end, so maybe there are other interesting questions for you. I will now introduce Julien CHLASTA from Biomeca. Hello, Julien. I let you the floor, and see you in 7-8 minutes.
Thank you.
I can see your presentation.
Yeah? Okay, thank you. So, hello everyone. My name is Julien CHLASTA. I'm CEO of Biomeca. First of all, thank you to Skinobs for giving us the opportunity to present our solutions. So our mission is to bring a biomechanical dimension to cosmetic claims. And today I will first introduce our company and expertise, and then focus on our innovation, the EasyStiff device. So let me briefly introduce who we are. Biomeca is based in Lyon, France. Our multidisciplinary team, biologists, physicists, and engineers, works directly on our projects, from studies design to data interpretation, to help you build more innovative and defensible claims. And importantly, everything is done in-house by the Biomeca team, no subcontracting, so you get speed, full traceability, and consistent quality. At Biomeca, our expertise cover hair, skin, nails, etc. For example, for hair, we study hair protection, characterization. For skin, we analyze bioprotection, tensor effect, anti-aging repair, and penetration. We also conduct assays such as traction force assay directly on the cells, wound healing, and cytotoxicity. So our approach extends from in vitro to ex vivo, answering reliable and translational results. Now let's take a closer look at the main mechanical properties of the skin. So we have elasticity, which allows the skin to return to its original shape after being stretched. Compressibility, the ability of the skin to be deformed under pressure. Viscosity, which describes its resistance to flow and internal frictions. Tensile strength, the capacity to win strength and stretching force without tearing. And finally, stiffness, which reflects how resistant the skin is to deformation. Altogether, these properties define the skin resilience, protection, and beauty. So skin is a multi-layered organ, and each layer has its own mechanical role. The stratum corneum is a strong barrier. The epidermis is responsible for renewal. The dermis provides tensile strength, and the hypodermis acts as a shock absorber. Understanding these roles is essential to link biological changes with product efficacy. So this is Assistive, our innovative device dedicated to skin biomechanics. Our philosophy is very simple: to bring biomechanics out of complex lab and make it accessible, fast, and reliable for cosmetic research and clinical studies. With Assistive, we are able to simplify a very technical approach while keeping all the precision and reproducibility required by international standards. This introduction slide reflects exactly what we want to deliver: clarity, innovation, and simplicity in skin biomechanics. Our goal is to make biomechanics simple and accessible. We developed a solution that can extract parameters from each skin layer in just one gesture. It is perfectly suitable for non-experts, so anyone can use it effectively. This is a real breakthrough. In only one measurement, we can capture biomechanical data across all skin layers, and this system analyzes raw data in less than 2 seconds, and results are available directly on the web interface. This means fast, easy, and powerful analysis. Here you can see the working principle. The device performs an indentation directly on the top of the skin and records the response. This indentation reveals the stiffness and elasticity of the different layer. This is non-invasive and precise way to measure skin mechanics. Beyond indentation, our system provides tomography stiffness. This means we can obtain stiffness profile at the stratum corneum, the epidermis, the dermis, and now the hypodermis. It's a unique way to visualize and quantify the skin's biomechanical behavior. To validate the Assistive system, we designed and carried out a clinical study on skin entrancing aging. The study was performed on the inner left arm, which is considered a protected area of the body, less exposed to external environment factors like sun or pollution. And this makes it particularly relevant for investigating natural intrinsic aging mechanisms. We include 47 healthy volunteers. This broad age range allowed us to capture the natural evolution of mechanic skin properties over time. Our measurements clearly showed that the skin stiffness increased with the age. On the left graph, you can see the global stiffness value gradually rising across our age groups. On the right, the result become even more interesting by separating the contribution of each skin layer: stratum corneum, epidermis, and dermis, and so we can observe how each compartment change over time. This demonstrates not only the ability of our device to measure global skin stiffness, but also to provide the layer-by-layer analysis, which is truly innovative compared to the conventional methods. The system is reproducible and repeatable, ISO compliant with a viability below 4%. EasyStiff allows us to connect biological modification with cosmetic claims. For example, cells differentiations linked to aging, lipid changes relate to moisturization, a collagen variation explain firmness, and pumping effect. This makes the data relevant for both science and marketing. With EasyStiff, the expectations are clear: fast measurement, simple use, even for non-experts, precise biomechanical data for each layer, and objective support for cosmetic products claims. And it's the solution that reveals the secrets of the skin. To conclude, Biomeca is a creator of solution in vitro to in vivo. With EasyStiff, we make biomechanics fast, simple, and accessible. Our innovation strengthens cosmetic claims with robust scientific data. So thank you very much for your attention.
Thank you very much, Julien, for this presentation of these innovative solutions to measure biomechanical properties of the skin. Just to remember our participants that the CQ from Pixience or the EasyStiff, you can find them again in the testing platform. Just filtering by instrumentation makers or by claims or what you like, or you can find also the Innov&Sea solutions, and also in the preclinical testing platform for ecotoxicity, marine ecotoxicity, you find the several specification of the Innov&Sea solutions. And you find also CIDP as a CRO and with their worldwide location. So now it's my turn. Julien, maybe you can just sorry, there is a question for Julien.
Okay.
Just one short answer, please. Martina Barberis Vignola asks you, in clinical studies, is it also applicable for in vitro tests like 3D tissue?
No, EasyStiff is designed for clinical studies on volunteers for the reconstructed 3D skin tissue. In this case, we complement our approach with atomic force microscopy, which allows us to measure the biomechanical properties of tissues with very high precision at the macro scale. So we have the solution to measure biomechanical properties on the tissue.
Okay, thank you very much. So now I will start with the testing trends at IFSCC. I will share my presentation. Blah, blah, blah. Okay. I hope everyone will see my presentation.
Yes, we see it.
Thank you very much, Gabriel. So let's start for a quick. You will get all the slides, so you can come back to the slide. They are quite full, but I will go through all this information. The IFSCC Congress was really great, and I wanted to show what are the latest trends and maybe what are the low signals of testing among all the information, among all the podium and the posters. So I will go through what we found at the IFSCC Congress regarding testing. Then I will give you Skinobs testing trends. And then we have developed Skinobs barometer about testing trends. So share with you, and I will give you just a very, very short summary of what we found as a main key drivers of testing of the testing field for cosmetics in the last part. So these are the exhibitors at the IFSCC Congress. So there was representation for clinical evaluation, preclinical assays, and device manufacturers. For the podium, so here my interest is only on testing. So this is a representation of what we found about the podium. And here they said that they receive more than 1,400 posters, and there were totally 700 posters. But regarding testing, we noticed 377. And here, these are the categories that have been presented by IFSCC. I did not change them, but I just noticed that sometimes we had, for example, on formulation topic, posters subject that was not directly related to formulation, but I think it was really a very, very difficult to sort all these posters in these categories, but that's what we found. And here, this is what we observe from the IFSCC Congress in the posters, through the posters and through the podium. I made this synthesis diagram because I wanted to show here in vitro ex vivo. What we saw the most was related to sophisticated models, 3D models with vascularization. We had a lot of presentation regarding innervated model and neurons. And here you can see there is a in-between area that is okay for in vitro, but okay for in vivo also. And we saw that neurosensory is for both, because now neurons are really, it's like a classic opportunity to test the ingredients and the finished products on these specific models. People spoke a lot about transcriptomics and microfluidic, and multiomics were on both sides, in vitro and in vivo. I thought that there was a specific interest for the dermal epidermal junction and all the mechanisms of action related to this part of the skin. On in vitro, we spoke about dynamic, about organ on chips. Also, it's very microfluidic, as I said, and cell shape and movement. That was my main point for in vitro. And for in vivo, I will say tone and wrinkles and holistic, of course, and structure of the membranes and inclusivity. Asian population. That's right. That I don't have the exact figures, but I think that the posters and the podium they were about half part presented by the Asian speakers and Asian scientists. They spoke a lot about gravitational force and saging, and there was a real aspect about instrumental measurement with a multimodal approach. Then this here, I synthesize the main topics that was discussed during these 3 days of scientific conferences. About longevity, aging clock, multifactorial skin aging process, sensitive skin, stress as a driver, and a lot of information about inflammaging, oxidation and glycation, pollution, of course. Photo projection was one of the main topic of the podium. So we spoke a lot about UV standard, and microbiome was also a main topic. So, but microbiome, the technology, also for aging, for acne, for UV mainly. And then radiance and facial translucency remain a real trends coming from Asia and traveling all over the world with I saw many interesting information about deplux de pores on posters. So that was the first one. The previous slide was about like a photo, and here we find more my insight about the testing trends, and what was overrepresented, on my opinion, was consumer. Why? Because of perceived efficacy, co-creation, regional adaptation, and cross-population. And there was a lot of posters about this topic. A big representation about skin, hair, scalp and hairs, but the hair was also a category, so it's normal that it was represented. And we can see neuroscience and desires. And I saw a very interesting podium from CPAN, and you can see the number, it's what you can find. And it was very, very interesting testing method to evaluate how we can answer the desire of consumer by using beauty routine. We can see also hyper personalization through 3D print, mask, and also specific personalization of products like Pro's offer. In silico prediction was there with IA, of course. I forgot IA, but IA, it's really everywhere. And we saw, as I said, sophisticated models. Microbiome was overrepresented, and clean beauty. And for what we miss, on my opinion, was testing trends or testing information about male, about menopausal. There was some, but I was quite disappointed. But even it's a real needs and request from the consumer. There was not a lot about nails. I saw only one poster about nail. Child, not that much, but it was about atopic dermatitis. And I will say that the African continent was really not there, unfortunately. And out of the box, I noticed that through the presentation of Julia, but just a message to take home is no worries. AI will not replace human, and AI is more artificial information than artificial intelligence. And it's until the next augmented intelligence, we have a brilliant tool to use every time. The second one was about with Nina Jablonski, and she take the point about the skin barrier, and the skin barrier as a second skin. So, and the third topic was the space exploration, because there was a specific roundtable about space exploration with many expert and scientific scientists. So if you want to test your products on in the space, you can do, and you can contact the European Center for Space, and they do. Okay, so let's go to the low signal in testing. I think that this is for testing. It's not It is not consumer or trends about what consumer like, but it's really on testing. The low signal is skin energy. I think eco-sustainability will increase in testing, even if there is no regulatory constraints for that, but people start to speak about more and more. Slow aging, hyper-personalized cosmetics and diagnostic. Also through conversational avatar, that is really new. Ethical, it's a low signal coming. Multi-pathway network, also local climatic conditions, and full spectrum photo protection for testing, because everyone speak about UVB, UVA, IR, blue light, and visible light. But I think that maybe the cosmetic, the beauty domain, the beauty industry is ready for testing in this field. And the fascia, this is I think a low signal of evaluation. Here it's synthesis of worldwide testing market. I will not comment because I do that for every post-show webinar. You find what inspired me in this field of testing market, and what are the main parameters that impact the testing. It can be testing technology, it can be beauty, cultural, and also political environment. These are the skin barometers coming from all the exchanges we have with users, with cosmetics brands, with experts, with consultants. And this is an overall way we design about, this is about testing field. Then here we have the main claims per continent, and here technological factors, how they influence each continent. And here you find that on our last white paper. So if you want to see it more deeply, you have the link, and you can find that on our cosmetic testing news website. So we speak about long term in terms of societal and scientific transformation, postmenopausal, medicalization, personalization, clean beauty, and microbiome. And about technological inspiration, we speak about neurocosmetic. Neurocosmetic was everywhere at the IFSCC. We spoke at home diagnostic also, and we had the opportunity to present a poster with Jean-Jacques Servant and Pascal Barrier for this, giving a panorama of what is possible. Also biomarkers and omics revolution, SPF, new standards from last year, and the boom of in vitro and in silico models. Thank you very much. This is our new interface and new redesign of the cosmetics testing news. It's free. We have more than 5,000 articles we share since 10 years, and now we have a great search engine. So it's really interesting when you search for any kind of word related to the beauty. Thank you very much. This webinar is recorded, and the replay will be on our YouTube channel. So thank you. I hope we will see you at Cosmetotest in March, and our next webinar will be around the Asian testing trends in October, and our next event will be in India and in Lyon, in Cosmetotest, and in Bangkok. Thank you very much. Now I will watch the question if there are any. I ask my colleague to come back with me and to end this session. So, um, okay. I have a question from Annick Foligné for Julie. Regarding the C-cube, is it possible to treat explants?
Yes, yes. Thank you for your question. It is possible to capture image of skin explant with C-cube and then make same measurement than on normal skin in vivo. It is more used for pigmentation and roughness actually by our customers.
Okay. Thank you very much. We have a question from Anna Toso. Thank you very much for your question, Anna. This is for Pauline from Innov&Sea. How do you manage the co-culture between cells and algae? I imagine that the cells are immortalized and can be frozen, but don't the microalgae in symbiosis suffer in this process? And is the co-culture cultivated with antibiotics or not?
That's a lot of questions. Yeah, thank you very much for this question. Yeah, I didn't really have time to go further on the technical aspects of the technology. Don't hesitate to send me an email or something to discuss this more after. Just to be quick, we don't use co-cultures of animals and zooxanthellae. We used two different cultures incubated in two different medium, based also, the both are based on seawater for sure, but there are two different cell cultures. We can use just one, or we can use the two if we want, but not in co-culture. About the immortalization, the animal cells can be frozen. Yes, we can cryopreserve them, but they are not immortalized. They are primary cell cultures. We don't cryopreserve the microalgae, in fact. I don't remember. Let me check the end of the question. Yes, we use antibiotics also, yeah, for in the cell medium. If you want to discuss this further, please don't hesitate to send me an email. And there's the last question about specific endpoints. Just to be quick, our test is currently using two specific endpoints, the viability and the growth for our routine test. But of course, we can use any kind of specific endpoints depending on what you want to see in the toxicity of the product. But our current endpoints are sufficient, are really very specific, and they're adaptable for any kind of product. So, but yes, you can use any gene expression or activity of any kind if you want. Thank you for the question.
Thank you very much, Pauline. Thank you very much, everyone. I think it's time. We have it was more than 1 hour, and we try to keep the time, but the time goes fast. Thank you very much to all our speakers to be there with us and share your expertise, because it's so important for the beauty industry to have science-based products to guarantee the consumer that everything is fine. So thank you very much. And we will be there for you anytime. And don't hesitate to ask your question, send me an email, or send directly an email to each speaker if you need more details. Thank you very much. Have a good day and see you soon. Bye-bye.